Hi, hello, good evening to all. Welcome to Unacademy YouTube class. Unacademy is one of the good platform to achieve the goal in your oncoming Karnataka CET. Myself, Murali PG. Today I am going to have the class on strategies for enhancement in food production chapter. Okay. Under this chapter, we are going to have the MCQ. So Unacademy is introduced a plus subscription best plan for Karnataka CET, and it is for Karnataka CET 2021 aspirants. It's a 12-month plan is there, 6-month plan is there, and 1-month plan is there. Okay, you can use it. So it is 12-month plan, 6-month plan, or 1-month plan. Okay. (12-month, 6-month, and 1-month plan is there, and it is with the live classes regarding with the MCQ, doubt clearance, and mega quiz will be there, followed with the MCQ. Then unlimited access is going to be there for this plus course, and you can have the presence of or use of this user code Murali853057 and get 10% off regarding this class. Okay, and secondly, the comprehensive batch, it was introduced from last 2 months, and where it is also coming under plus course. Complete syllabus is going to be covered, tips and tricks by top educators, topic wise practice questions, and full length mock test will be there. So you can have this one also. So it will going to enhance you with respect to our Karnataka CET 2021. Use my referral code MuraliPG853057 and get 10% off with respect to price value. And third one is the champion batch. So champion batch is the third one, where PCM, PCMB or PCB is there, and it was started on 15th of October. Enroll your name for this class also and get a good idea about our Karnataka CET. Then a newly introduced daily practice questions, T20 free practice questions test for Karnataka CET, started on November 2nd. Enroll for the test, attempt it live, and let's crack Karnataka CET together. So use this link and get all the information regarding with my classes. And I am taking special classes. I am taking our plus courses under comprehensive batch. So if you want more, you just subscribe for the plus course under comprehensive batch or champion batch, and you will get all the things regarding with the Karnataka CET 2021, my dear students. Okay, by this brief introduction, now I am heading towards discussion of class questions under strategies for enhancement in food production chapter. Okay, so today's class is strategies for enhancement in food production. Okay, so let us start our question answer discussion. The first question under this chapter, it is what is or expand moint. Expand moint. What is mean by moint? So multiple ovulation embryo transfer, multiple ovulation endosperm transfer, multiple ovulation embryo transfer, none of this. So out of this question options, the right answer is multiple ovulation embryo transfer. So it is moint is important to increase the herd size of cattle in a short duration of time. Here multiple ovules are going to be produced because of superovulation by the administration of ova producing hormone in large quantity. So as a result, we can have the increase of herd with respect to cattle in the short duration of time. Okay, so that is what the answer for this question number 1. And second question, which among the following is marine fish with respect to strategies under aquaculture? That is musa, sardine, mackerel and cutla. So the right answer is C, cutla. Cutla is one of the marine fish observed in the ocean. Third one, the process by which crop plants are enriched with certain desirable nutrients. Certain desirable nutrient is called as what? Crop plants are enriched. Crop plants are incorporated with, enriched with certain desirable nutrients to human being for human consumption. Which is that one? So the answer for the third one is the biofortification. Biofortification is the right answer. Yes, Manohar T, namaste, welcome to the class, good evening. So third question, answer is B, biofortification. Okay, then fourth one, tryptophan and lysine are what? Whether they are lipid or carbohydrate, protein or lipids. Once again, so here option tryptophan and lysine for fourth one, it is the protein. Okay, it is the protein. Protein is the right answer. So D is not needed, repeated thing. Okay, next identify the process of plant breeding from the diagram shown below. So here I given one diagram. What is the indication of this diagram? What is the major step of this diagram observed in the case of plant breeding? So fifth one, B is the right answer. Artificial cross pollination. It is the process by which the emasculation is carried out, where unwanted stamens are going to be taken out and bagging of that flower to avoid the contamination from unwanted pollen grains. So it is the process in the case of our plant breeding phenomena to get desired character in the oncoming generation. So that is what artificial cross pollination, in that bagging and tagging phenomena. So emasculation, bagging and tagging. So here it is the emasculation. Okay, process is the emasculation. And next question number 6. Norman E. Borlaug or Norman Borlaug, the father of Green Revolution, has developed a highly recognized and appreciated cultivating variety of what? Which variety he was developed? Whether it is wheat, rice, onion, sugarcane. Of course, all are very important crop plants. In that, Norman E. Borlaug is called is considered as the father of Green Revolution. The answer is wheat. Wheat is going to have its own importance in the whole world. In India, North India is a major crop, isn't it? So wheat is one of the important crop plants, and this wheat, it was developed by this person, that is Norman E. Borlaug. Next question number 7. Am I right? Yeah. Which one of the following help in absorption of phosphorus from soil by plants? So here mutualism is there between the microorganism and the plant root. So which is that one? Anabina, Glomus, Rhizobium and Frankia. It is the Glomus species. So if you take if you take Glomus, it is one of the important mineral assimilation type of fungus. So it will provide this phosphorus from the soil into the plant. Plant will provide nutrition and accommodation to this Glomus species. Okay, so that is the answer. Yes, Devesh, welcome to the class. Rakesh, welcome to the class. Good evening to all. So we are now in the 7th question. So right answer is Glomus species. Next question. Selection of homozygous plant is what? What is that? It is the mixed selection, mass selection, pure line selection, none of these. So 8th question, it is called pure line selection. So homozygous, isn't it? Homozygous refers to having identical. So homozygous plants are selected, means it is a pure line selection where particular identical characters are going to be there in that plant. Okay, am I audible clearly? Rakesh, Devesh, Manohar? Okay, next question. An organism used as biofertilizer for raising soybean crop is which one? So here nostoc, Azotobacter, Azospirillum and Rhizobium. So with respect to our soybean crop, to increase the yield, it is the Rhizobium. So responsible with respect to fixation of nitrogen, isn't it? Next question number 10. Question number 10. In tissue culture medium, the embryos formed from pollen grains is due to which culture? The production of embryo or embryoid from the pollen grain is going to be there under tissue culture. In that, which kind of tissue culture is implemented here? Double fertilization, organogenesis, cellular totipotency, test tube culture. It is the cellular totipotency. Totipotency is the phenomena by which one cell is going to develop the entire organism. That is the capacity of that cell. So the cell which shows totipotency, cellular totipotency, is good for considered as explant for the tissue culture. So cellular totipotency is observed in the case of development of embryoid from the pollen grains. So answer is C for the 10th question. Yes, Rakesh, you are right. And 11th one. Hybrid vigor is mostly due to what? What is the reason behind with respect to obtaining of hybrid vigor? So pure line is there with respect to homozygosity, isn't it? So homozygosity of pure character means it is for pure line. Superiority of all the genes means good characters. Then heterozygosity and mixing up of cytoplasm of the male with that of female. Cytoplasm mixing is not right, but heterozygosity is the right answer. Hybrid vigor is the mix of both characters. So that is the heterozygosity. Okay, heterozygosity is the right answer to obtaining with respect to or to obtain the hybrid vigor. Then main objective of production or use of herbicide resistant genetically modified crop is to see the main objective, main purpose of production or use of herbicide resistance genetically modified plant or crop plant is for what purpose? It is for to reduce the herbicide accumulation in food articles for health safety. So reduction with respect to accumulation of chemical herbicide in the food articles or in the food chain on the regard of health safety. So to overcome that one, we are going to have the particular modification in the case of plant. So it is called genetically modified crop, GM crop, where herbicide resistance or use of herbicide resistance or production of that kind of character in that crop. So that is the main reason is A, reduce the herbicide accumulation in food articles. So we can reduce the incoming of that unwanted chemicals in the food chain. Next, 13th one. In which crops is the method of mass selection applied? Mass selection. It is cross-pollinated crop. In the cross-pollinated crop, we can observe the method of mass selection. Okay, method of mass selection. Got it? So if you have any doubt, Devesh, Rakesh or Manohar, you can ask, okay, with respect to any question, answering, etc. Next, plants provide protection from fungal disease by producing which one? 14th one, it is phytoalexin, the chemical which is going to be produced or chemical which is going to be utilized. So plant provide protection from fungal disease by the producing phytoalexin kind of chemicals in their body, means in the plant body. Next, sterilization of tissue culture medium is done by which method? So here, mixing the medium with the antifungal agent, keeping the media at minus 200 degrees Celsius, autoclaving of medium at 1200 degrees Celsius, and filtering the medium through fine sieve. The right answer is autoclaving. Autoclaving of medium at 1200. Sorry, it is not 1200. One second, I will going to correct it now. It's actually 120 degrees Celsius. Okay, 120 degrees Celsius. Sorry, 120 degrees Celsius. So autoclave is the process. It is a technique where the boiling of that media above 100 degrees Celsius to kill all the microorganisms. Okay, so that is what the sterilization phenomenon which is needed. So right answer is C for 15th one. 16th one, the problem of necrosis and gradual senescence while performing tissue culture can be overcome by problem of necrosis and gradual senescence, gradual aging, isn't it? During the time of tissue culture can be overcome by which method? That is subculture. So if you are going to use same culture media with the same kind of explant for long duration of time, then there will be the necrosis and gradual senescence, gradual aging of that part of plant body which is utilized, which is going to be utilized in the case of tissue culture. So we have to do subculture. So new tissues are going to be generated, so we can overcome the necrosis and gradual senescence with respect to that particular tissue culture explant. Okay, so answer is 16. For 16, A is the right answer. Okay, next question number 17. Plant tissue culture media can be solid, semi-solid, liquid, all of this. What about the nature of that plant tissue culture? It is all of this. It may be either solid or liquid or semi-solid or all of this. So here you can observe all of this is the right answer. Okay, yes, for Shishika, welcome to the class. For 16th one, A is the right answer. Okay, next one, the main purpose of using UV radiations in plant tissue culture, in the plant tissue culture media preparation or in that area where we are doing the plant tissue culture process with respect to laminar airflow chamber, why we have to use the UV radiation before the commencement of that plant tissue culture process? That is just for disinfecting or disinfection. So this disinfection should be there, otherwise what happens, there may be contamination with respect to fungal spores or by this fungus or bacteria or any kind of microorganism. So to overcome that, before the commencement of entire process of plant tissue culture technique, in the case of laminar airflow chamber under that tissue culture lab, we are going to apply UV radiation for about nearly about 30 minutes, etc. So as a result, we can eradicate all the infectious agent or the contaminating agent in that locality of laminar airflow chamber. Then match the following and choose the correct combination from the option given. Here I given some of the plant material or animal material: honey, bee venom, propolis and beeswax. So here the answer with respect to second one, the utilization of that one. So 19th one, A is the right answer. A is the right answer, means here for 18th one, for 18th one you are right, Shishika, that is B. And for 19th one, the right answer is A, refers to honey is artificial sweetener. B, bee venom, it is utilized in the treatment of rheumatoid arthritis, is a genetic disease, autoimmune disease, isn't it? And propolis, it is antiseptic and antibiotic property with respect to that compound. And beeswax is used in the cosmetic creams, etc. So right answer is A for 19th one. Then 20th one, the best way to obtain bacteria and viruses free plants through tissue culture is which one? So best way to obtain bacteria and viruses free plant. So 20th one, it is shoot apex or meristem culture. Meristematic tissues are going to be utilized, or shoot apex with the meristematic tissue is going to be utilized for the purpose to do this tissue culture as explant, because in the meristematic region cells division is very fast than that of bacteria or virus multiplication. So shoot apex is with the meristem, and meristematic tissues are going to be utilized as explant with respect to this tissue culture. So even though the other part of the plant body may have the bacteria or viral infection, but the meristematic tissues are never going to have this virus or bacterial infection. So because the cell division of meristematic region is faster than that of cell division in the case of virus and bacteria, that is the reason behind that one. So 20th one, A is the right answer. 21st, which one of the following product of apiculture is used in the cosmetics and polishes? Apiculture means honeybee culture, isn't it? In that which is the right one? So honey, oil, royal jelly, wax. It is the beeswax. The wax production produced from the honeybees are going to be utilized in cosmetics and polishes under carpentry work, etc. So one of the economic byproduct of honeybee is in honeybees. Then 22nd question. 22nd question. Okay. More than 70% of livestock population is in which nation? Livestock population, total domesticated animals. So here answer is India and China. India and China are the highly populated countries of the world, and about 70% of the livestock is still present in these two nations. Okay. The agriculture sector of India employs how much percentage of total population? So it is once again 62% of population is directly or indirectly depending upon agriculture sector in our nation. Yes, Rakesh, you are right. For 22nd question, D is the right answer. Okay. So the agriculture sector of India employs about 62% of the population. Yes, Sheshika, you are right. For 23rd question, it is D. Then 24th, 33% of India's GDP, gross domestic product, comes from which sector? So 23rd, so it is 24th, yeah, 24th, it is by the agriculture sector, because 60, about in the previous question I said no, about 62% of population depending upon agriculture. So as a result here, about 33% of India's GDP, it is coming from agriculture sector itself. That is the importance of agriculture in our nation. So strategies for enhancement in food production, how to enhance, how to increase the production by different strategies for applied biology. That is what the subject, isn't it? And the 25th question, it is a collection of all the alleles of all the genes of a crop plant is called what? All the alleles responsible for expression of particular character with respect to all the genes of a crop plant is called what? The technical term, it is the germplasm collection. Germplasm collection. Okay, 25th question, answer is A is the right answer. Got it? Yeah. So totally 30 questions are there. So we are in the 26th question. Yes, Manoj, Sheshika, Rakesh, Manohar, Devesh. Yes, 26th question. What is this? System of rotating crops with the legume or grass pasture to improve soil structure and fertility is called what? What is that phenomenon? 26th question, it is called shifting agriculture, strip farming, lay farming and counter farming. So the correct option is C, it is a lay farming. So system of rotating crops with a legume or grass pasture. So leguminous plants are going to be grown in the paddy field or in the wheat field, so to increase the nitrogen quality of the soil, because leguminous plants are going to have the home, the root of the leguminous plant are going to have the home for nitrogen fixing bacteria, or if you take grass pastures. So as a result of this rotation of this crop, main crop with the leguminous plant, in the next season for the main crop they are going to get nitrogen very easily. So for this purpose in agriculture sector, usually to improve the soil structure and fertility, the leguminous plants are going to be grown in between the duration of main crop growth. Okay, that is called lay farming phenomenon. Then 27, which of the following enhances or induces fusion of protoplast? Fusion of protoplast. So protoplast fusion ಮಾಡಬೇಕಿದ್ರೆ ಏನು ಮಾಡಬೇಕು? Which is the chemical? It is the polyethylene glycol and sodium nitrate treatment should be there. So protoplasm will go into fuse. So protoplast refers to the plant cell without cell wall. So that fusion of protoplast will be there. Protoplast culture is there. So it is done by the application of polyethylene glycol and sodium nitrate. Then 28th question. A technique of micropropagation is what? Which is that type of micropropagation? In that somatic hybridization, somatic embryogenesis, protoplast fusion, embryo rescue. So for 28, it is the somatic hybridization, somatic embryogenesis. So technique of micropropagation, somatic cells are going to be utilized under this production of embryos. It is a technique of micropropagation. Then the process of induced breeding of fish is called as what? Induced breeding of fish, it is called hypophysation. So for this the fish is going to be administered with the hormones. So superovulation methodology. So it is called hypophyseation, to increase the number of offspring in least time gap. So hypophyseation, where fish is going to have induced with the hormonal injections or hormonal influence. So for 29th, A is the right answer. Manoj, for 28, B is the right answer. You are right. Okay. For 29, A is the right answer. So in the case of moit also, in the first question I said multiple ovulation embryo transfer. There also the inducing of hormone is there, isn't it? So to produce multiple ovules in a single reproductive cycle. Then last question, who is the father of white revolution in India? Green revolution, I already said, Norman E. Borlaug. With respect to white revolution, with respect to dairy sector, with respect to milk and milk production, milk byproduct, etc., who is the father of that section? It is Verghese Kurian, who established the Amul dairy at Gujarat. Okay, Amul dairy at Gujarat. So that is Verghese Kurian is the person considered as white revolution in India. So one of the well-known person under this category. Okay, so by this I completed 30 questions, and here if you want more you can subscribe for plus under Unacademy. So best plan for Karnataka CET 2021 aspirants with the following 3 important type of plans, and in this plus class you will get a good theory, doubt clear session and MCQ, and followed with the mega quiz. You can have the unlimited access to this class and use my referral code and get 10% off with respect to this price value. And secondly, comprehensive batch, complete syllabus is going to be covered, tips and tricks by top educators, plus educators will going to take the classes, topic wise practice questions and full mock test for individual chapters. And myself, I am taking this one from last 2 months. Along with me, the 3 other educators will going to take their respective topics. Once again, use my referral code and get 10% off. So my referral code is Murali PG853057, and here the champion batch for Karnataka CET 2021, where PCM, PCMB, PCB is there, and it was started on 15th of October, and enroll your name for this one also. Okay, and daily T20 free practice test for Karnataka CET, it was started on November 2nd. Enroll for the test, attempt it live. Let's crack Karnataka CET together. So by joining these courses, you can enhance your power to face the questions in the oncoming Karnataka CET. So good night to all. I am going to wind up this class here. Thank you one and all. Okay.